So, so a little bit of my background. I have PhD in health and biomedical science, but also I've done a postdoc at the University of Cape Town focusing on high throughput microbiome data analysis. So I've been working on microbiome for some time now, and I'm really happy to talk on the aspect of public engagement and advocacy in research. And for that matter, I'll use microbiome research as a case while communicating this aspect of genomic and genetic research in Tanzania. And here is an outline of my talk. I'll give a background on what microbiome is, and then I'll take you through the current microbiome research at a cutting edge. In this aspect, I'll try to pinpoint or to advocates and call for public engagement. In this aspect, I want to introduce this idea, especially for Tanzanian researcher, because these great types of research has been going elsewhere. So I want them to understand the aspect of this so that we can use it in our country context. The third part, I'll highlight the opportunities that are available related to genomic research in Tanzania. And then in the last part, there'll be a question and answer session. So microbiome is defined as the total genetic material of microbes. These are bacteria, fungi, and virus that are found on and inside the human body. The total of these fungi, bacteria, and the virus, genetic material are what makes the microbiome. And this dates back to year 2007 when the Human Microbiome Project, this was an NIH US-based initiative, when they wanted to improve the knowledge on the microbial flora that are involved on human health and disease. So they wanted to know the microbial flora that are involved in human health and disease. And because of that, several studies have come up reporting that in human, microbiome consists of between 10 to 100 trillion microbial cells that live on and inside the body, the majority of which are bacteria which are found on the gut. So we have around 10 to 10 trillion microbial cells that live on and inside our body. Remember, the human body, on average, they have around 30 trillion cells, but inside and outside our body, we have the bacteria or microbial cell up to 100 trillion. Genome-wise, it has been also reported that human has an average 22,000 genes, some are in 20 to 25,000 genes, while the microbes found on and in our body has around 3.3 million genes. That's like we are carrying 150 times genes of other living organisms a part of ourselves. So probably if you are to be rational, you would say probably we are microbes and we are not human beings because they constitute a larger part of the gene that we carry. It has, because of that, it's now rational to say that humans are thought as a composite of human cell and microbial cell. And also the human genetics is an aggregate of human genome and the microbiome because we constitute a lot of these from the microbes. Likewise, if you think of the metabolic features, it's now judged as a blend of human and the microbial traits. We carry all this information within ourselves. Unfortunately, this has been realized later in the year 2007. So if you think of genes and the cells and the metabolic features, these are the major factors actually that constitute the disease and health condition of an individual. So if we keep ignoring these microbes, you can think of how the consequence will be because genetically they outweigh in terms of genes. And also if you think of total cells, we have so many cells of these microbes than ourselves. And metabolically, they contribute a lot on our health. So some researchers have gone far even saying that microbiome is considered as a forgotten organ that we can't ignore anymore. Because of these findings, we can no longer ignore these microbes that constitute a larger part of ourselves because there is a lot of evidence that any changes in microbial profile that we have in and outside of our body is actually leading to high susceptibility to diseases. It's because of the absence of some of the microbes or presence of some of the microbes that we get the disease condition. So it's very important that we know the role of these ones. And because of that, several studies have been reported or working on the microbiome research. If you're able to see that figure there, this is the proportion of the microbiome research, microbiome research. A lot has been done on gut and also host metabolism, immune system, diet, and inflammation. And if you check from this plot, actually started around the year 2006 to 7. These are the number of publications that has been working on microbiome research. And most importantly, you can see, if you check on your left, these are linked to diseases, yeah? So if you check, for instance, infections, we have a lot of publication related to infections. Here, on average, was around 3,000 publications per year on infection. But also there is some correlation between the microbiome profile and the cancer diseases, also the obesity, also the diabetes, also the liver diseases. So all these kind of diseases, in a way, they somehow defined or contributed by the changes in pattern of the microbiome that we carry. So studies have shown these ones. And for instance, if you are to retrieve this from year 2008, you could get around 40,000 publications related to microbiome. But I did just the day before yesterday, I could get around 624,000 publications stating about the microbiome and relation to diseases. So this is a great kind of research, is a cutting edge. A lot of people out there, they are doing this kind of research. And I'm happy to say that this, we as Tanzanian or researchers, we have this opportunity because this one has been done elsewhere. We can do the same in our country. If you think, for instance, of cancer, we have so many cancer cases nowadays. In most of cases, we think of the radiations or the types of whatever exposed some chemicals that are causing to cancer. But researchers has shown that we get some metabolites. When we get some of the food, we get some metabolites after they're causing cancer. And these metabolites, normally they are not acted properly because we miss some of the microbes that are very important. So these are the opportunities that we could do. Other studies have shown that the microbiome profile is changing with the age, especially if you think of at the 0 age or immediately after birth, we see we have very little microbes or 0 microbes. But with days, you see the number of microbes is increasing, and this is a function of the environment or the parent where the baby was born, and the nature of birth. If you think of cesarean and home birth, they have different pattern of these microbes. But with the age, you see a different profile of microbes can be found. You see firmicutes there, maybe most abundant, maybe bacteroides or actinobacteria. So the type of microbes that are present in an individual is a function of several variables. It could be food, could be the environment where someone is living, or the life pattern, lifestyle. All these are contributing to the microbiome profile, and it's very important to know them because they have some consequences that we get. Again, if you think of the immediate after birth, you see here on the left, the individual variation is very high at birth, and because here we have the microbes that are in most cases defined by the lifestyle of the parent or the mother, and the type of birth, and also the environment where the baby was born. But with the age, we see this individual variation is becoming very negligible or very small. And in this case, because these individuals who are in the same environment, then are exposed to the same kind of microbes, then they'll have more or less similar, if you see, of the contribution of these. And again, the diversity goes high because now you'll be getting different species of microbes in or inside your body. All these are very important when it comes to research because they contribute different metabolic function, different disease condition, or otherwise. Studies also has shown that microbiome is a function of diet. So whatever you eat, sometimes in most cases define also the microbes that we'll get. For instance, if someone is based on a plant-based type of food, where there is high fiber, low fat, and low protein, you will see the gut microbiome will constitute high diversity of the microbes. For instance, you see here we have 5 genera, we have 5 bacteria genera here, and because of that, we get also an increase in short chain fatty acid, equal, and dimethylamine. These metabolites, actually, they are beneficial to health. And if you check from literature and some other studies, these microbes, they contribute to prevent the formation of cancer, but also the cardiovascular diseases. But if someone is based on animal types of food, I can see here the different types of beef and the like here, you'll have a higher increase of these types of bacteria with only 3 bacteria genera here, but there will be an increased trimethylamine oxide, precursor, a NOx compound. Unfortunately, research studies have reported that most of these metabolites, they contribute to the formation of cancer. Most of them, they are mutagenic or carcinogenic, and also they do something to cardiovascular diseases. So if I were to recommend, maybe we should focus on plant-based food material rather than animal-based material because of these consequences. But the thing here is this has been done elsewhere. We could do the same in a Tanzania context to see what pattern do we get. We could also do the sampling of our colleagues who are suffering from cancer, for instance, try to see what are the microbiome profile they have. And it could be probably they miss some of the microbes that are beneficial here. That's why they're developing the cancer disease. Then we could do the transfer or fecal transplant of good types of microbes to these ones. Yeah. So to highlight of what would be the opportunity in Tanzania, we have so many areas. If you think of this as community of scientists in Tanzania who are doing microbiome, we really need to push around and make the ball rolling. What is being done out of the country, we could also do in our country because we are all human beings and we could also benefit on this kind of research. For instance, if you think of the infant, for instance, you could check the microbiome profile based on the type of birth, whether cesarean or normal birth, because some studies have shown that cesarean birth has some effect to the microbiome profile. You get very low diversity of bacteria to child born through cesarean because they miss some of the microbes that are normally found through the birth canal. So, and this is associated with development of immunity to the children, and also a challenge when it comes to susceptibility to diseases once they grow. You could check also the breastfed versus formula-fed microbiome. Studies have shown that those children who are breastfed, they benefit because they get some microbes that are beneficial and they help in the metabolism of their food. And that way they benefit when they grow. Again, you could check the preterm birth versus vaginal microbiome. I've been working on one of the research where they're trying to establish the microbial profile or the vaginal microbiome profile as a function of preterm birth. There are some bacteria that are associated with preterm birth. We could be experiencing a preterm birth in our country because those mothers, maybe they be having some bacteria that actually should not be there. They could be contributing to that. So that's a potential area that has not been done in our country. You could also check nasopharyngeal microbiome to see how these affect the kids, especially pneumonia diseases. You could also check how the microbiome is changing with diet. Some cohort could be taking a certain kind of diet, then we do the monitoring of the microbiome profile. Again, you could also check on the microbiome and the allergies. Some people are susceptible to several types of allergy. And this could be because they are missing some of the microbes that are beneficial, or they're having some microbes that actually should be there. Again, in the adult, you could check the cancer's patient, those without the cancer, to check the microbiome profile. And again, the microbiome profile has been reported in the brain-gut axis. Actually, they help us to think. They define us the way we think, the microbiome. Probably that's why when you get hungry, you feel angry or you become nervous. Some studies have shown that the brain, the way our brain works, is a function of the microbe that we have in our gut. So that's also an area. There is also an aspect of obesity as a function of microbiome. Some people are obese because they miss some of the microbes that are beneficial. So if we know this from our generation or from our population, we could do a way to transfer those beneficial bacteria so that they can also recover from the diseases. Again, some studies have been reported, relation of sickle cell disease in the microbiome, also diabetic in the microbiome, or also the microbiome and HIV. For instance, there is one study that we did that was based on sample collected in Zimbabwe and in Malawi. It was reported that for children that have been taking antiretroviral for a longer period of time, normally they generated a microbiome profile which is as good as like those who are not suffering from HIV. So if we know this information, our community could really benefit. Again, you could check the intestinal bowel disease, like you compare how these microbiome are related to the disease that are happening in our gut. Again, you could check this microbiome against the cardiovascular diseases. In most cases nowadays, we think So once they have scarosis, it's because they take a higher level of fat, but it could be attributed by the bacteria that there is a miss or they have some that are not beneficial. So we could take a cohort of people who are suffering from cardiovascular disease and then try to monitor their microbiome profile. Later on, we can come up with a solution to advise our people on how to handle this one. Again, we have a lot of infectious diseases, and this could be contributed by the absence of some of the bacteria that in a way they prevent colonization of those pathogenic bacteria. So these are very critical areas that we could research. I'm really excited to put this forward, and if we can communicate, especially to our scientists who are working in the medical field, these are potential areas that we could work together. And if we do that, then we'll be able to do an effective diagnosis of diseases. And sometimes we might be doing wrong diagnosis. I'm not a medical doctor, but there is a chance that we do a wrong diagnosis. Someone is suffering certain disease, you take your sample, you culture in the laboratory, then you say this is the bacteria causing the disease. Actually, studies have shown that if you take the sample from the patient, you do the culture in the laboratory, you will miss 99% of the microbes that are present in that sample. The reason is most of the microbes are not yet known. And since we don't know them, we don't know their nutritional requirement for them to grow. So we don't provide the appropriate environment for them to germinate in our laboratory. So we miss them. We might be making a very wrong diagnosis. But with the molecular approach, you take the sample, you start the DNA, then from the DNA, you can amplify all the target genes that are causing disease, or the whole genome of the microbes that are present in there. From there, you can get a true representation of the microbial profile that you have, then a proper diagnosis of the disease. So with microbiome, again, you can come up with a personalized medicine because different people, they have different lifestyle, they have different microbial profile because they eat different type of food, they have different, and these contribute to the microbiome profile that they have. So this kind of generic medication actually is something that we need to do away with time. So we can also make new discoveries because this area is still very virgin, especially in our countries, or in other countries is somehow has gone to a higher level. So what our move is our advocacy is to bring the medical field in Tanzania, whereby we can have a cohort of individuals who are doing the clinical studies to study the microbiome profile of different diseases. And from there, once you have the cohort, you then generate a database. In this case, you can have a sequence or metadata information related to those individuals. And from there, you do the bioinformatic analysis. From the bioinformatic analysis, then the researcher or the doctor can do a better therapeutic decision based on the finding that they get here. And here you can do the prescription, whether you give a probiotic or prebiotic. Probiotics, these are a kind of food that are having live or living microbes. And the prebiotics, these are types of food material that actually they stimulate growth of microbes in the individual. Then once you give this probiotic to the individual, then he or she will be able to recover from the microbe that we are missing. Or you can do a fecal material transplant. You introduce the pure isolate of those missing bacteria to that individual. Then you do the monitoring to see whether the pattern is changing or not. And when you do the monitoring, then you come up with a profile, whether this is recovered or yet to be recovered. This profile also is kept in the database so that our researchers or our medical doctors can use this reference to do a diagnosis. So this is the approach that would be very appropriate instead of just taking a sample, do the culturing, because in so doing, we miss a lot of information that would help us to do a proper diagnosis of the disease. So because the topic's about human genetics, but microbiome goes beyond the human genetics. Microbiome also can be exercised in agriculture. For instance, this is a part of the study that I did during my PhD. There is one dump site that I wanted to see the profile of microbes that are present in that dump site. Unfortunately, in that dump site, you see these trucks of floating the garbage. By then, that dump site was free for anybody to get there, so it was not closed. You could see a lot of garbage from the medical, from the market, from the homes, but also you could see these food animals scavenging on the dump sites. And these are not wild animals. Later on, these are slaughtered, then they are eaten by human beings. So I wanted to see the profile of microbes that are present here. Because this is a zoonotic nature. We have the human here, we have the animals, we have the polluted environment. So this study, I was able to capture a lot of microbes that some of them are really reported to be pathogenic through extraction of the DNA. Then you do amplification of target genes that are universal for all the bacteria, and then establish a phylogenetic to see what's the link between these microbes that are present in that area, and what could be the potential risk for those people who are working the dump site. Actually, I also did a comparison of the profile between the pigs that are found on the dump site. These pigs were there throughout their life, but also I did the monitoring some of the pigs that were raised indoors, far away from the dump site. Then I transferred them to the dump site. I did the monitoring for 1 month to see what could be the profile. Interestingly, I could see the profile here. As you see here, the green here are the indoor pigs and the free-range pigs in red. Interesting, some of the microbes here that are found in the scavenging pigs, for instance, bifidobacterium, Crucibacter, and Enterococcus. These are the bacteria actually that are used in some cases to make probiotics, or you make yogurt using these bifidobacteria. These are beneficial bacteria. So it's very likely that these bacteria are present, selectively present in the pigs scavenging the dump site, and they help those pigs to be safe from the other pathogenic bacteria, because they inhibit they generate the so-called colonization resistance. They resist the pathogen bacteria to prosper from these animals that are always in the dump sites. And we could use the same approach to either to harvest these types of microbes and use them for medication, or to purify these and use in human for treatment. Again, you can use microbiome approach if you want to study the environment where you do the agriculture. For instance, I'm working with one of the research in our group. They use called the push-pull technology for farming, where you'll have some plant that actually facilitate to grow some of the microbes in the soil. And those microbes, they help to generate nutrients that are beneficial to the plant. So you can only know the profile of microbes that are present in a given environment or in a given plant if you use the molecular approach. If you do the culturing, as I've said, the area, you'll miss most of them. So the microbiome can be used in a broad area. It's a really great opportunity for researchers to get into this type of research, because our community, our society, our country will benefit from it. So how to get started? If you are very new and excited on this one, you need to identify the potential area of genomic research. And then you identify the collaborators, international and the local collaborators. In this case, Tanzania Human Genetics can assist you, because we have a good network of people who are working with genetic disease or genomic diseases or whatever. So you should also consider take advantage of the competence and capacity that we have locally. And that's why we have this kind of symposium, because we know who is doing what and where is here available and what are the skills that we have locally. In some cases, you may find it takes a lot of time to find help from somebody out of the country. By knowing the locals, that would be really beneficial for us as a country. Again, we suggested that you can make a collaborative grant proposal, and this should be multidisciplinary to involve all the experts that can have some contribution to the team, because diversity is very important when it comes to a very competitive type of grant. Again, engage the public on what you are searching on, because as you have seen there, we need to have a cohort. If the public, they don't know what you're doing, it's likely that they won't be able to participate in the research. We need to engage the public on what we are doing and tell them the benefit that we'll get out of it, and that's really critical. So that's an overview of what I wanted to say. There is an opportunity for us to do in our country. We have the network, we have this champion from Tanzania Human Genetics. We can really use this chance to get into it. And our university, especially those who are working on medical field and those who are working in agriculture, there is a potential opportunity to use genomic approach, because in so doing, you get more informative information on whatever hypothesis you are working on. Thank you very much. That's the end of my talk. I would really appreciate to the organizers for this symposium. They've made a lot of effort, the time to make this possible. I appreciate for them, especially for my employer for giving me an opportunity to participate on this. Also, the Tanzania Society of Human Genetics and all participants for your presence. I'm happy that you have been part of us. Thank you very much. And I'm inviting you if you have any question or comment or suggestion. Thank you.
Thank you very much, Dr. Samson, for such a lively talk. I hope participants have questions or comments. Can you type questions and comments in the chat box? We'll be able to let Dr. Samson to respond to one question or comment, and then he will continue to engage in the chat box with further question and comments. Then we move to the next speaker.
Okay.
So any questions, comments from the audience? I'll be looking in my chat box. And as we continue with other speakers, you don't have to wait for the talk to end to type a question or a comment or any remark. You can already start typing. So when the speaker is done with the talk, it will be easier to address the question.
So I'll be available in case of any question. I'll be in position to respond.
Hello.
Yeah.
Yes.
Thanks, Dr. Samson.
Yeah.
It was a very good presentation. So I have a question.
Yes.
So the current situation is most of these researches, especially on the microbiome, depends on the data generations, and that means the things like sequence data or the metagenomics data. So what is the current situation, the current condition in Tanzania, especially on the things like the capability on generating this data and the competence, especially of the people, bioinformaticians, on analyzing this data? What is the current situation in your opinion?
Well, thank you for a great question. In terms of capacity, this is one of the objectives of society. We want to identify all researchers or experts that are available in the country, and what area of research are they able to work on. Frankly speaking, we have the experts already in the country. The only challenge that we had earlier, we are not having a kind of association or a network that can put us together. For instance, in terms of microbiome, I know we have some facilities at the KCIR, KCMC University here. There they have a Illumina MySeq platform where they can do the sequencing. And in terms of data analysis, for instance, I'm also expert on data analysis. I did the bioinformatics. I can do any kind of analysis related to microbiome. And again, we are now moving forward to identifying other researchers that are present in the countries. And so for sure, if we were to do the microbiome, whether we are using 16S or targeted sequencing approach, we have the competence to do the processing of the data. Just to mention, when I did my PhD, I generated a lot of sequence data. I generated 30 million sequence data. And it was a challenge by then on how to process them. I relied to some guys who are out of the country. At some point, it was hard for me to rely on them. I have to venture on my own way. And through that hard way, actually, I managed to process my own data, and I've attended several training on that. And because of that, I was given an opportunity to do the postdoc at one of the reputable computational biology at the University of Cape Town, where I did further skill building on how to do the microbiome data processing. And again, we have the network of H3 Africa, which is helping a lot in terms of data analysis, especially the microbiome. And we offer a lot of training online. For instance, we have the IBT training that ended, I think it was October or November this year. We have some experts in Tanzania that did the learning of that training. And for sure, we can do this data analysis. The only thing we need to pull our energy together, and so that once we collaborate, then we can make one thing a whole. So the competence we have. Thank you.